Hello everyone, this is Sukanya. Today I am going to discuss about enzymes involved in gene cloning. A gene cloning is a process in which a gene of interest is located and copied, a cloning out of all the DNA extract from an organism. And what is a cloning? A cloning refers to process under to create copies of DNA fragments, and the activities of cloning enzymes consist of ligases, ketones and proteins of DNA ligase. Example used in joining DNA molecules together with, while DNA primases replace DNA using DNA replication. DNA cloning. DNA cloning is the act of making copies of a single DNA, and DNA cloning can be provided a pure sample of an individual gene, separating all the other genes that can be normal shares of the cells. Once a DNA gene is identified, clones can be used in many areas of industry and biomedical researches nowadays. And enzymes involved in gene cloning. As above have said about gene cloning is a process in which gene of interest that are 10 types and nucleases, polymerases, ligase, DNA, RNAs, ketones and kinases, polymerases. These are the main roles which can be involved in DNA cloning. Nucleases. A nuclease is an enzyme capable of cloning and phosphodiester bonds between the nucleotide subunits of nucleic acids and exonuclease and enzymes that work by cleaving and nucleotides one off at the time of end of polynucleotide chain, and this phosphoester bonds can be either 3,1 or 5,1 prime ends occurs, and nucleases are classified into two types based on its functions: exonuclease and endonucleases. And exonuclease is cuts with phosphodiester bonds towards ends, and it doesn't cleave to DNA strands with the terminal 3,1 OH groups because they are blocked with excited groups. And endonuclease it cuts with the phosphodiester bond within a polynucleotide chain. It consists of exonuclease which cleave phosphodiester bonds at the end of the polynucleotide chain. This is about endonucleases, and this polymerase process cuts a piece of DNA. The newly polymerized molecule is template's original partner strand. Zero DNA polymerases use magnesium ions can be cofactors. Polymerases are four types: ONA polymerases, RNA polymerases, 437 polymerases, T10th polymerases. These are the four types of polymerases. These are five types of DNA polymerases: poly 1, poly 2, poly 3, poly 4, poly 5. These are the four types of DNA polymerases. There are 15 unknown eukaryote DNA polymerases. These are 15 known eukaryote polymerases as shown below. And as you can see picture of DNA polymerases of replication, double strand DNA, wood chains of formation of ANA, AME DNA, single strand DNA. Ligases. Ligases are special type of enzyme that in the single primer, single strand DNA polymerizes molecules in which simple words strands that can have double strand break. Purification of DNA ligase is used in DNA cloning to join DNA molecules together. These four types of ligases: DNA ligase 2, DNA ligase 3, DNA ligase 4. These are the four types of ligases. DNA ligases that can be DNA of lagging strand after DNA polymerase 1 have removed the DNA primer from the fragments, and DNA ligases 2 are alternative slicing from the DNA ligase found in non-dividing cells and complex with DNA replication protein. And DNA ligase 4 it catalyzes the final step in non-end joining DNA double strand breaking repair path. These are the four main roles DNA ligases. Transferases. These enzymes add nucleotides with 3-strand DNA strand, three type of DNA strands. They are called as terminal, DNases. DNases are three types. ODNases 1, DNases 2, and these enzymes degrades aminoases. DNases are used to degrade DNA. These enzymes perform random degradation because they are not specified to recognition sites. And as you can see a picture, NCL DNases, as I have said above, key and kinases. Kinases are the enzymes which adds phosphate group at 5N. Kinases are also called as polynucleated kinases. Kinases are which can be involved in phosphodiester bonds are the enzymes which remove phosphate group at 5N, and they are also called as alkaline phosphates. As you can see above, a protein kinases are involved in this ATP, ADP. Restriction enzymes. These are also called as molecular scissors, and these enzymes are specified towards dsDNA. These are clones under the family of nucleases. These are discovered in bacteria of 1962. They cut DNases at or nearby specific recognition sites. These are also called as recognition sites. Recognition endonuclease are three types. They are type 1 IRE, type of 3 RNA, and all recognition endonuclease follows two rules. For secular DNA, N rule, and linear DNA, N4-1 rule. And applications of gene cloning. These are the applications which can be done in gene cloning activities as production of protein from clone genes and production of recombination, pharmaceutical, medical sciences, and synthesis of human growth hormones in E. coli, recognition of vaccines, and this can be done applications of DNA cloning. And thank you.